Welcome to the Grandmaster Series. I'm your host, Jim Scott, sometimes holly jolly, but usually just cheerful and smiling, because I just like to smile. Smiling's my favorite. Before we get started, there are a few things I'd like to share. Anything discussed which is publicly available will be shared in the video summary below. URLs, other references, things of that nature. If you have any questions during the video, you can submit them through the chat. We'll try to provide answers throughout and at the end of the episode. And finally, please be sure to subscribe to our channel to be notified of upcoming videos in the Grandmaster series. This is how we keep track of how popular this series is and how well prepared we should be to continue servicing our audience. I'd like to introduce the team before we get started. First off, we have Boyan Tungas. He is a competitive machine learning modeler at NVIDIA. He holds a PhD in physics from UIUC and an MS and BS from Stanford. He is a 2x Kaggle Grandmaster and has been ranked in the top 20 for all categories. Next, we have Kazuki Onodera-san. He's been a Kaggle competition Grandmaster since 2019, and he's had 5 top 2 competition rankings. Next, we have Amit Erdem. He's currently a senior data scientist at NVIDIA with a background in computer engineering and artificial intelligence. Next, we have Dr. Christoph Henkel, a PhD in mathematics with a focus on probability theory and stochastic processes, and he is a senior deep learning scientist at NVIDIA. He is a 3x Kaggle Grandmaster. Next up, we have Jean-Francois Puget. He holds a PhD in machine learning and has published over 70 scientific papers in peer-reviewed conferences and journals. He is a 2x Kaggle Grandmaster. And finally, for this episode, we have Chris Diat. He's a senior data scientist at NVIDIA. Chris has a PhD in computational science and mathematics with a thesis on optimized parallel processing. And Chris is a 4x Kaggle Grandmaster. So thanks everybody for joining today. I have to say, I am jubilant. I am so excited to bring you today's episode of the Grandmaster Series because everyone knows the best way to spread Christmas cheer is by singing loud for all to hear. Today, the Kaggle Grandmasters of NVIDIA will be singing their favorite Christmas carols. Wait. What? That's not what we're doing today? Well, I dressed up special for this and got my microphone out. Special for singing. Come on. Seriously? Okay, fine. Okay, let me find the right notes. All right. On today's episode, we'll be speaking with Kaggle Grandmasters of NVIDIA in an attempt to distill their experience into actionable and repeatable learnings from which you, our esteemed viewers, can benefit. Today, we'll specifically be covering the Kaggle competition for COVID-19 vaccine degradation prediction. This is a topic that is likely very close to everyone in the community, given the ramifications of the COVID-19 pandemic. All of us have been impacted in some way by this terrible virus, and hopefully we can all join together to obliterate this pandemic. Now that the stage is set, I'd like to hand it over to Boyan. Boyan, take it away.
Thank you, Jim, and thank you everyone for joining in. As Jim mentioned, today's episode will be focusing on a very special Kaggle computation in which we were trying to predict the degradation rate for mRNA vaccine molecules. And there will be a few very different things that we're going to talk about in this one. We will, again, cover, as Jim has already done, the teammates on the Kaggle, NVIDIA's Kaggle Grandmasters team. Then we will just give a general overview about this mRNA project and how Kaggle came to play a very important role inside of it. Then we'll introduce solutions from some of the top teams from NVIDIA's side. Second place with Kazuki Onodera, 17th place with Ahmed Erdem, 20th place with Chris and Jean-Francois, and there will be actually additional 33rd place by Chris Diot. And then we'll have a brief Q&A session after that. So as already mentioned, we have a nice, strong team of Kaggle Grandmasters at NVIDIA. And in various capacities, 6 out of 9 of us were actually involved with this competition. I myself have had a very different involvement in this competition than all the other ones that I've been part of, and I'm going to briefly talk about that aspect, which is something I think very interesting to everyone. These are the top ranking teams from Kaggle, from NVIDIA Kaggle team and their placement. As you can see, there were several teams that came in very strongly. They would probably have even done better if the competition ran longer, but this was again, very special competition at very short time scale that did not allow for more sophisticated models than what we've been able to come up with. So as everyone knows, we are in the middle of a COVID pandemic that's been taking a lot of attention internationally, domestically, everywhere. It's been impacting the world in many different ways, and there are strong efforts now that are already bearing fruit to bring a new vaccine to the market. And this one is very different from all the other ones that have generally been used. The effort was on this particular vaccine effort is mostly concentrated on trying to come up with more stable vaccines, not really developing the vaccine itself. The effort was led by Riju Das Lab at Stanford University, who was one of the people who contacted me about helping with setting up the competition. My role was kind of dual role, being a Kaggle Grandmaster, knowing about Kaggle competition, as well as being part of the NVIDIA team. And I was providing some counseling before the competition launched. The competition was, again, on the very short time scale. There was a real need to kind of set up a competition really fast and have results available to scientific community in kind of record time. So it was very different. The competition leveraged two major crowdsourcing platforms, Kaggle, which probably all of you are familiar with, and the other one is Eterna, which is an online game where competitors are tasked with solving RNA folding puzzles. So this is one of the I think this is the first time ever that two different crowdsourcing platforms were actually involved in a solution to a major scientific challenge. So as we all know, the COVID-19 is still ravaging economy, people's lives, and health. It's the worst pandemic in over a century. It has a very high fatality rate, debilitating and a very debilitating economic and social impact. And there's a need for as much of an effort to be done to actually try to mitigate all of this. And short of developing full herd immunity naturally, the best possible solution for it would be to develop a vaccine. The problem with traditional vaccines is that they have a very long development timelines, and usually it's about 18 months to 2 years, which was not a very feasible to actually get an immediate impact on the COVID vaccine. So there was a need for a very different approach, and the approach that has already been kind of tried and tested over the decade, but never really at this scale, was to develop mRNA vaccines. Now, the difference between RNA vaccines and traditional vaccine is that instead of having a weakened microbe, which you have with a traditional vaccine, with mRNA, you try to get RNA into the cells of the body so it could create the proteins that are associated with that particular microbe. So it's kind of bypasses several levels of development that otherwise would take way too long to do. Problem with mRNA and RNA in general is it's a very fragile molecule. It very easily degrades, especially at the room temperature. So developing RNA vaccines that would be robust and easy to use in general population has been a major challenge. The two vaccines from Moderna and Pfizer that are right now used require really low temperature refrigeration and are very infeasible to be used across the globe. So there's an attempt to create RNA molecules that are more stable than the ones that are currently being used, and that effort is still ongoing. There was a hope that maybe even these solutions can be used with the current vaccines, but that was way too optimistic, and probably the next it will have to wait for the next level of vaccination. Now, RNA molecules, I'll briefly talk about them again in the next slide, but essentially very long, complex molecules that consist of 4 base pairs, which are adenine, cytosine, uracil, and guanine. So just these 4 pairs can create a very complex, even 3-dimensional shapes that have still not been well understood how they form. So again, this effort to create more robust RNA molecules was spearheaded by Riju Das lab at Stanford University, which is a basic biophysics of RNA molecule labs. Kaggle is an online community of science data science practitioners that tries to solve machine learning problems, many of which have a very important impact on the real world. Eterna, again, is this website that I would urge you to check out if you haven't done so yet, where it's very interesting how pretty much anyone can join and try to solve these very complex real-world scientific problems that pertain to the folding of RNA. So here's a few things about how a competition was organized. Again, I was approached by CEO of Kaggle as well as Riju, who needed some kind of advice about how to set up this competition in a short period of time. So they really need to kind of jumpstart it and go through it in much faster way than usually Kaggle competitions are done. Machine learning competitions are, despite what it may appear, are really hard to set up. There are many pitfalls, and even the professionals from Kaggle and other platforms have hard time anticipating all of them. So usually, I think in Kaggle, there's a long preparation period of months, if not half a year, from the time when the organization approaches Kaggle to actually have a functioning competition. For this particular one, we had to actually kind of speed it up, get it within the weeks out. And the big question was, can you really do justice to a competition in such a short period of time? I personally was very skeptical about it, but decided to kind of jump in and see how much I can help. And some of my suggestions and some of my insights came to be important for the way that competition was run. So there was initially an attempt to create stable RNA molecules before the vaccines go in production, but that didn't really pan out. But still, this information is being used for further development. One of the challenges in setting up a proper competition is how do you prepare your data? You come up with a lot of raw data. Some of it is relevant for the organization when it's using it, but may not be so relevant for the competition itself. Initially, I was very skeptical that you could make a very good model because there was only about 3,000 molecules that they had the data on, but turned out each molecule has like 100 or so base pairs or base amino acid nitrites that were each one of them could be viewed as a single data point. So it wasn't as bad as I initially feared. There was a big discussion of how to decide on train test split. Also how to use clean data. There was a lot of data that had a lot of noise, and initially everyone was leaning to kind of filter it out, just use the very clean data for the competition. But I prevailed on them and said, like, no, just give them all the data. They're Kagglers, they know how to deal with it. They'll figure out even how to use this dirty data to actually get the best possible model. And I think that was one of the crucial reasons why competition was in the end successful, because there was just enough data to create models that can generalize. There's always a choice of metric. Some metrics can really be skewed by outliers. And initially, the lab was using this R-squared score, which was kind of very hard to work with. And for continuous data, usually RMSE or log RMSE are better choices, and we settled on RMSE as a default metric. One of the biggest challenges was, keeping my friends and colleagues in dark about this, because I didn't want to create any kind of situation where even the appearance of impropriety of kind of giving them special insights about it would come to play. And that was very important, since I believe strongly that many of them would be finishing very highly ranked, as we have seen in the end that did transpire. And then, like, for me, the hardest part was actually recusing myself from the competition, because I really like to be part of every single competition. And in this one, not only did I recuse myself as a player, I decided to recuse myself also as an organizer, because, again, didn't want to have any kind of appearance of impropriety when my teammates would eventually win. So competition itself had about 3,000 RNA sequences, and each one was of length 107. The ground truth was provided for first 68 bases, because it was very hard to get it for the other ones. There were also 629 of these sequences. 629 were in public test set, and there were additional 3,005 sequences for which, at the beginning of the competition, there was no ground truth information. So again, this is a very pure machine learning kind of environment, where even the organizers don't have information about the final test set. All of the public test set, again, was pure, very high quality, very well balanced, in order to make a public leaderboard pretty consistent and reliable. Every Kaggle competition eventually has a big shakeup, and initially, because of some kind of encoding error, they seem to have had a huge shakeup. But eventually, when it was rescored, turned out the shakeup was fairly minimal, and that, to me, is one of the indicators whether Kaggle competition is successful or not. If there's no major shakeup, and you can be you have pretty reliable local validation, train validation, test validation, and private validation. Again, it was a very short competition, ran from September 10th to October 6th. People were kind of scrambling to get good models, but it was very impressive what kind of models they came up with. In terms of competition, it was nominally a tabular data competition, because all the data was presented as a CSV file, plus additional files that encoded parts of the 3D structure. So that's the general kind of setup of competition. And at this point, I'm going to hand it over to Kazuki, who will talk about his second place solution. Kazuki, you're on.
Yes. The 10 is my slide starts.
You need to go to the next slide?
Yes.
Okay.
Yes, please.
All right. Let me just a second. All right.
Yes. Now I'm introducing our second place solution of this competition. This team consists of me, Kazuki Onodera, and Kazuki Fujikawa. We used LSTM, GRU, and GNN. Okay, please go on to the next slide. Here is our pipeline. In this competition, the data augmentation was the key. So we augmented the data using ONI. I will show the example in next slide. After the augmentation, we created LSTM, GRU, and GNN. And then generate probability with stacking using XGBoost. The result of the stacking got second place on public and third place on private. After the stacking, we tried pseudo-leveling with cream prediction. We use a test sample with cream probability of 0.75 or higher as pseudo-level. we use test sample, After that, use XGBoost again for stacking and got first place on public and the second place on private. So this was our final submission. Okay, please go on to the next page. We augmented the data using ERNI. ERNI receives the structure and then generates the wide variety of BPS. I think all teams in Gold Zone use this library. Okay, next page. In this section, I'm introducing LSTM and the GRU model. We embedded sequence structure and predicted loop type, and then stacked multiple VPS in the channel direction. Next page, please. In this section, I'm introducing GNN model. Sequence and the predicted loop type were encoded as one hot, and then create edges using structure. The VPS generated by ERNI were converted into distance matrix. Okay, go on to the next page. Now I will show you how to avoid overfitting with Gaussian noise. Let's say LSTM1 is trained by pseudo-level data. When we do stacking, it causes overfitting because it's drastic prediction. So we added Gaussian noise and prevented from overfitting. The noise range came from readable. Okay, go on to the next page. And in this section, I'm presenting simulating a real world. We came up with simulating LV. Train sequence length is 107 and level is 68. But private test sequence length is 130 and level is 91. We need to know how degrade our model in private test. So we decided to slice the sequence in line with the ratio of sequence. After this experiment, we know our models will perform better in private tests. Okay, next page. This is the result of stacking with XGBoost. The yellow square stands for the result of stacking, and the red circle stands for mean averaging. The other stands for the single model. So now you can see how stacking is better than mean averaging. Both public and private. This is the end of my slides. Thank you for listening.
Thanks, Kazuki. I will present about 17-place solution. I'm not going to present the whole solution, but some ideas from it. In this competition, I teamed up with Rob. He trained and assembled a lot of models, which gave us significant boost. I want to thank him for that. While he was working on multiple models, I was trying to improve single model performance. And today I will explain the things that improve single model performance. I am not going to give all the details because it is similar architecture, I think, with other competitors. It was repeated bidirectional GRU, attention, 1D convolution, and dropout. I repeated this 3 times over the data. And the things that was novel for this competition was patch dropout and CME's architecture. Next slide, please. So the dropout layers are commonly used. We all know them. And standard dropouts, they randomly drop some features. In other words, they set them to 0. But they are not very preferred in sequence problems. Spatial dropout is usually better in sequence problems, because with regular dropouts, adjacent nodes can get missing information from each other, which was very similar information, so they can do it as workaround. With spatial dropout, they don't have this opportunity. It drops whole feature map on the data. But then the model totally missed this information, which could be very crucial. Therefore, I have implemented a custom dropout. I named it as patch dropout. Maybe it exists before, but I didn't know its name, so therefore I call it patch dropout. It's something between standard dropout and spatial dropout. It doesn't drop whole feature map, but it drops some patches, and most of missing information sharing is prevented between adjacent nodes still, but information from far nodes are allowed because they are probably different. This gave us some good improvement on the model accuracy. Next slide, please. One back. There was one slide in between. Yeah, this one. Thanks. Yeah, as Kazuki mentioned, there was RNA package. With this package, given a base sequence, we were able to generate structure, predicted loop type, base pair probability matrices for different temperatures. So then I decided to create this data for different temperatures, and I wanted my model to utilize the similarities between them or differences between them. For this, I created a CME's architecture. Same data is fed into identical backbones. Why identical? Because they share weights. Therefore, model needs twice less parameters. It means less parameters means less overfitting, and they can learn from each other. Then output of these backbone models are concatenated and fed into a hidden layer. This hidden layer can then do the interactions between these different data. So that's it from my side. I leave it to Christoph.
Yeah. Hi, thank you, Ahmed. So I want to present Jean-François and myself's solution. We had a strong team of Jean-François, myself, and Philip, who works at H2O.ai. Our solution is a bagging of a single model, which is a 3-level stacked bidirectional GRU, plus an inception block of a convolution. And we also added the base pairing using concatenation of the related nodes and their embedding. Next slide, please. But before coming in more details into our solution, I want to give a quick overview about graph neural networks and their basics, because I guess it makes our solutions more understandable. So what's more commonly used in machine learning competitions are recurrent neural network and convolutional neural networks, which you can see on the left side. So recurrent neural networks are often used for data that has a sequential format or sequential properties, like a sentence, for example, and then you propagate from one word to another word. Whereas convolutional neural networks are often used for images, where the idea is that given a small or given an image, like you can see in this example, which is just an image of a digit number 7, which you can show as a 27 by 27 grid. You then use different convolutional filters which go over this image, and you aggregate the information given in the whole image in different levels until you end up with one embedding vector or one feature vector that represents this image, and then you use that feature vector for whatever task you have at hand, often a simple classification. Now, with graphs, it's a little bit more different because you have neither a sequential nor like a grid kind of structure, but you have like a graph structure where a graph is nothing other than just a collection of nodes and edges, as you can see in the image here explained. So and you want to somehow interact between the different nodes along edges. And to add a little bit of terminology, the graph, which I show on the left, can be represented, or the edges can be represented as an edge matrix, which is a matrix consisting of zeros and ones, which just shows which nodes are connected to which. And then the idea of neural networks on these graphs is that given node and edge features, you basically exchange information between nodes along edges, and you update information within nodes and edges. Next slide, please. Now, coming to this competition, a simple example how to formulate this competition as a graph neural network problem is that you consider the bases as nodes and basically derive edges from the base pair probability matrix. So a simple example would be to encode the information for each base, namely the base itself, which is one-hot encoded, the structure which the base belongs to, the loop type the base belongs to. You encode that in a feature vector. Then you use an RNN approach to propagate sequentially the information between each nodes, and you end up with an updated feature vector for each node. And then you use the base pair matrix, which has continuous values between 0 and 1. And you binarize this matrix using some threshold to result in like a binary edge matrix. And then use this edge matrix to propagate information between the nodes, and you end up with an updated state of each base, which basically carries the information of the whole mRNA sequence. And then this updated state is then used to predict our targets. So that's a GNN on one slide. And with that, I would give over to Jean-François, who will present our solution in more detail.
Thank you, Christophe. So as Christophe said, we actually started with a generic graph neural network where each base is linked by edges to the previous and next in the sequence, and also to distant base via the BPS matrix. But performance was not great. And the reason is that the sequence formed by the RNA sequence is very strong, and using RNN is better. So we treated all the edges corresponding to the sequence via a GRU. RNN. We also tried LSTM, which was a bit weaker. Blending the two was good, but we lacked time. Our solution was developed in a very short time. Christophe and Philippe joined like 4 days beforehand, and I started like 2 weeks, and so we went quick. So we started with GRU, we stacked it, and then performance was better, but not perfect. And then, of course, we could add the graph component with distant base, and I will come to it, but still, we thought the baseline should be better. And then we started looking at this as a 1D equivalent of an image, a CNN for 2D images. So the GRU would be like the convolutions, the pooling layer. And then we needed convolution level. So we added convolution level at each level in the stack, and we got a model that was surprisingly close to Armet model. It's funny. The order of our levels were, if we swapped one submodule, we would get the same model. So it was surprising. A 3-level stack with a GRU and then convolutional networks. The convolution block, we tried several variants, and the one that worked best was similar to efficient net, so with a squeeze and excitation and skip connections. And we had good. We were like 400 until the last morning of the competition. And then, so next slide, please. No, you skip one. Yes, thanks. We had the hunch that GRU was not modeling correctly the structure of the molecule. So if there was, for instance, a big loop, if you have a big loop in the molecule, where the loop is connected to the rest is fragile. And there was no way, because you have a lot of weights on one side, and so we thought GRU was not capturing it. And fortunately, someone shared what is similar to an inception block, so with convolution, with increasing kernel size. So we just added it. Instead of having one convolution block, we have a stack of convolutions for each GRU level. I give the settings. And actually, there is a formula in PyTorch. I don't know for Keras TensorFlow, but it depends on the detail. But you have a formula so that for a convolution, 2 times the padding must be equal to dilatation times kernel size minus 1. So playing with that, these numbers perform well. But this is good, but we are still treating this as a sequence. So if you remember Christophe's slide, we are not really looking at the graph. So the second piece was to look at additional edges. Next slide, please. Okay, so this is, you see the picture. It's a simplified view of an actual molecule. So you see the sequence starting from the end labeled 5. You have pairs. They are linked to 2 neighbors, so it's a sequence. And here you see the sequence moving sideways and going back, and you see pairs, additional pairs. So these are additional edges, and we need to propagate information. So we could have implemented a generic, and we did actually implement a generic graph convolution, but this is a very special graph. bases are connected to one or two edges along the sequence, and then at most one pair. So you have at most 3 edges per node. So instead of a generic convolution, we just added a way to share the information across the pairing. So what we do is, after one level of GRU and convolution, we get a new sequence of embeddings for each node. Then we just permute the sequence to get the pairs. So for instance, this one would get a void pair, but this one would get the embedding of the paired sequence. So I don't know what index it is, but it is somewhere further along the sequence. This one would get the embedding of its pair, etc. This one would have no embedding because it's not paired. Then this one would get this embedding. So we get the sequence of embeddings of pairs. And for all these, for instance, this second sequence is empty. And we just concatenate. And we have a 0 embedding, or I don't know if it was a learned one or a fixed 0 for all the pairs that were not embedded. So just concatenation after permutation. And then we enter the next level. And this was quite effective because in the end, our model, a single model, finished 20. So it's not the best reported in the competition, but close to the best single model. And I really believe simplicity is very good in general and also in Kaggle, because a simpler model, as Ahmed, you said it, means you have less weights, and therefore you probably overfit less. So personally, I always try at equivalent performance to get the simplest model. And with this, I believe I will let Chris present his solution.
Thank you. So first, I'd like to congratulate my coworkers. This competition had 1,600 teams competing from around the world, and these are teams filled with professionals and experts in the field. So Kazuki's finished in 2nd place, and Ahmed in 17th, and Christophe, Jean-Francois, and Philip in 20th. Those are excellent models. So here's my solution. It finished 33rd place. Similar to other teams, I built a variety of models. I used recurrent neural networks, GRU, LSTM. I used graph neural networks. I used autoencoders and convolutional neural networks and attention modules. And using a variety of models is good because it helps deal with the noise that Bojan mentioned, which is in the labels and the targets. And then also there was a difference between the test and the trained data. So also using a ensemble of a variety of models helps the solution generalize. One of the things I did differently from other teams is how I dealt with uncertainty. So, so the data that was given to us had uncertainty in it. So we were given a sequence of nucleotides, which are these A, C, G, U. And so, the sequence is just a straight line. And then we don't really know what the molecule looks like in real life, but what it'll do is it folds on itself. So, the A's are generally attracted to the U's, and the G's are attracted to the C's. So a straight line will basically, it'll fold, and the bases will get paired together. So Kaggle provided us with the minimum free energy structure, and that's displayed here in the top left. So this is a one example molecule in the test data set. You could see the nucleotides are colored, the A, C, G, and U, and then the picture in the top left is the structure that Kaggle provided. But see, that structure is not necessarily what really happens in real life, and we need to feed these structures into our models. So the way I dealt with uncertainty is I considered the different structures that may actually occur. So if you look at the picture on the top left, I've identified with a number 1 and number 2, but those pairs of A and U, they're actually attracted to each other. So in real life, they might actually snap together, and then we'll actually have the molecule that's displayed in the middle. Now, the picture in the middle, I've changed the color coding. I've now, to help you out here, basically I've colored each base showing how likely it is to pair. So blue means the base is very likely to pair with another base, and yellow means it's unlikely. So you can see that, generally all the blues will make those long fingers. So the minimum free energy structure is a very high probability molecule, but you can see that other things might connect. And then in the middle picture, if you look at the 3, 4, and 5, those G's and C's, they might actually attract each other and fold into the molecule on the right. So in real life, we're not really sure, which form this molecule will take. It can take one of those three. So what I had done was, I took all the data in this competition, and then I generated the 3 most probable molecules. And here I've displayed the 3 for a test molecule. And this tripled the amount of trained data and tripled the amount of test data. And then I took all my models, and this trick was efficient because I didn't have to build new models. I can just take the new test data and then just train my models with the different versions of the molecules. And then all my models predicted the different versions of the test molecules. And then in the end, I had 100 different combinations of training data and prediction on test. And then I just averaged all of those 100 combinations. And then those are my final predictions.
All right. Thank you all. Thank you for your wonderful presentations and very insightful models that you built. I'll hand it over back to Jim.
Well, thank you very much, guys. That was really great. Thanks for sharing all the details on this competition and how you all end up with these best-of-breed solutions. So I've got a few questions I'd like to ask, and this is for anybody who'd like to jump in and provide an answer. So do I understand correctly that all the solutions submitted for this competition had to be made publicly available? And if so, where would people go to find these?
I believe that all the medal or gold medal solutions, or at least all the prize solutions, were supposed to be open sourced and provided with sharing license. If you go to the discussion section for the competition, most of the people who have open sourced their solutions would post links there. There are a few Kaggle notebooks as well, where most of the solutions from the top teams have been shared. So those would be 2 places to look for them.
So the next question I've got is, this is some really interesting stuff. It seems broadly applicable. I'm curious, is this something that this approach could be used for other vaccines?
Yeah, indeed, there are many other vaccines that could be potentially turned into RNA vaccines. So far, RNA vaccines have not been widely used because, as you've seen, that they're very unstable. But if this COVID vaccines proves can be stabilized, then this similar approach can be used for other vaccines, including the seasonal flu vaccine. So there's actually really great hope that seasonal flu vaccine can be even more effective. Right now, seasonal flu vaccine is developed, I think, 18 to 24 months in advance, based on the best guess what that season's main pathogens will be. But if this process can be sped up, we could have potentially much more effective flu vaccines.
Great. So do any of you see any follow-up competitions on Kaggle based on the results of this competition?
So I've been, again, involved with organizing this competition, and there's definitely interest to provide more data for both, on Kaggle as the platform, or potentially organize another competition down the road. I think this one was, again, more kind of hastily done that everyone would have liked. I believe that if a more gentle timeline can be given, we can actually potentially have even a better competition.
Interesting. And for the next question that I've got to kind of piggyback on the previous couple. And these questions are for anyone that would like to provide an answer. Are there any implications for other RNA-based projects aside from vaccines?
I think there's a lot that can be done with RNA molecules. They're very, if you know the biology of a cell, RNA molecules are the ones that tell ribosomes how to create proteins, and proteins pretty much build everything that's a living tissue. So there's a lot of more that we don't understand about RNA than we do about some other big biomolecules. As many of you might have heard, Google has come up with a very powerful solution for protein folding called AlphaFold. Currently, there's no similar efforts to do something with RNA molecules. This Eterna platform lets players create potential candidates for the folding of the RNA, but there's no ab initio deep learning solution, and that could be a potentially very interesting project.
Did anybody else have any thoughts on that? I'll take that as a no. All right. So
one thing that you have to know about generally about Kagglers and Kaggle competitions, none of us are really domain experts by a long shot, and all we really care about is data and how data is structured. So like all these additional things are nice icing on the cake, but we really don't know much beyond that.
Fantastic.
I would add something that in this competition and in a previous competition where we were asked to predict some other molecular property, Kagglers were first to introduce some technique. So using graph neural networks to predict molecular properties. It was first done on Kaggle a year ago in another competition, and it was reused here. And I believe Kaggle is often under-recognized in academia as a source of advances on how to model a given problem. And Kazuki's model and other medal-winning solution in this competition should really be looked at by academia.
Great feedback. So the next question that I have is, as I was listening to this, I was kind of curious. So aside from people who are very dedicated to the problem domain of data science, people who are experts in the field in general, are there any ways for people to be able to get involved in this? So I'm thinking of like the folding at home projects or other things like that. Are there any ways for people who are less experienced to be able to participate in this larger overarching area?
So yeah, there are two ways. You could go back to Kaggle itself and take a look at the data, take a look at the solutions. If you believe you can build even better solutions, go ahead, try it, build upon the ones that have been open source. If you're successful with that, please contact either people at the Reju Das lab at Stanford or me or someone else, and we would be happy to take a look at it. Another opportunities, again, to get involved with Eterna and start playing there. There you don't even have to have any computer knowledge or coding knowledge or data science knowledge. Being kind of inquisitive and able to visualize how molecules could potentially fold is all that you really need to do.
That sounds pretty fun. We'll make sure that link is in the notes below for everybody.
I'll add to that. I'll kind of reiterate what Bojan said. I just want to point out to everybody that every Kaggle competition, once it ends, it's still open for the public to build models and then submit the predictions and get a score. So, it's not like the competition's over and done. So, if you're interested in graph neural networks, you can hop in, you can read a lot of the discussions, you can copy a lot of the code from the public notebooks, and you can try, if you have an idea, maybe how to improve it, you can try to build a model and submit it. And then also, Jean-Francois mentioned an older competition, which was another predicting molecules. But once again, if you want to do some work with graph neural networks, you can hop into that competition, which was about a year ago. Again, build some models and you can submit it and get a score. So I just want to point that out to people that every old competition on Kaggle, you could practice and build a model and get a score still.
Excellent. That's really great for people to know. I'm sure that a lot of people are not fully aware of that already. So in our next episode, we're going to be covering the topic of NLP, which will be what I like to call kind of like a super episode, because there are going to be 4 different competitions that will be included and discussed. Could any of you share a little teaser for what our viewers might expect from that episode?
Yeah, so I would like the next episode to be a little more educational and give a brief overview of what happened in the last 1-2 years in NLP, because actually there happened a lot. And you can also see the evolution at the Kaggle competitions. So we will cover Kaggle competitions from like 12 months. So the last 12 months, I would say. And we had really great results there. And those competitions tackle a variety of problems, from detection of toxicity in Wikipedia comments, over how to do transfer learning between different languages, over how to do question answering. So a variety of problems, and yeah, really interesting next episode, I would say.
Fantastic. Well, that is the end of all the questions that I've got. I would like to personally thank Boyan, Kazuki, Christophe, Ahmet, Jean-Francois, and Chris very much for spending time speaking with us today. For myself and all the viewers, I'm sure they, much like myself, are feeling very enlightened at this moment. To all the viewers, thanks so much for spending part of your day with us and taking the time out of your otherwise busy schedule to enjoy this episode of the Grandmaster Series. For all my guests today, I'm the smiley, happy, joyous, seasonally frightful Jim Scott. Have a great day, and may all your data science be accelerated.